Welcome to this biochemistry lecture on the citric acid cycle. This is obviously a very complex subject. It's at the heart of our catabolic pathways right after glycolysis. It goes glycolysis, pyruvate, acetyl-CoA, and pyruvate and acetyl-CoA are the feeders into the citric acid cycle, which we've talked about. And you can see in this chart, that's where the entrance exists into the citric acid cycle. Just for reference, the citric acid cycle is called the Krebs cycle. It's also called the TCA cycle or tricarboxylic acid cycle. It's called any number of things. This chart gives, there's 10 steps in this cycle, and we have all the products, reactants. This is a good reference point for you when you're studying this this cycle. There's, again, products, reactants, enzymes, cofactors, everything's in this little chart right here. And we will go into detail in all of them. All right, so to start off with, oxaloacetate is converted to citrate. In this reaction, it's a condensation reaction of oxaloacetate with acetyl-CoA. It's a high-energy thioester hydrolyzation. The enzyme is citrate synthase, which you see in these two pictures up here. The hydrolysis provides the energy to drive step one. Oxaloacetate is going to bind to the smaller domain of the citrate synthase, and that's going to cause the domain is going to tweak a little bit. And you can see the tweaking in the difference between the closed and the open, and it causes oxaloacetate binding site. It seals it in there and closes the conformation of this homodimer. And you can see that, again, between the two states in the picture. The conformational change not only prevents solvent from reaching the substrate that's bound, but it also generates acetyl-CoA binding site to open. And this presence of the open and closed form allows citrate synthase to have what we, as you might recall, is an ordered sequential kinetics. And so, anyway, so that's what happens in this reaction. You can see the reaction at the bottom. Oxaloacetate on the far left is converted to citric-CoA. There's several regulators of this step. It's the first step in this reaction, and so it's an important regulator. Their regulators are obviously the substrates. There's acetyl-CoA and oxaloacetate. There's also controlled by the NADH and citrate, as well as ATP. So all those three regulate the first step in this reaction. Step two is done by aconitase. It's where we take citrate and we make isocitrate. Citrate and cis aconitate, and is an intermediate. But the final step goes from citrate to isocitrate through a intermediate called cis aconitate. And again, aconitate is a dehydration and then a rehydration. It's catalytically enzymed again by aconitase. So interestingly enough, citrate and aconitate are achiral. Isocitrate is chiral and has 2 stereocenters and 4 stereoisomers. So aconitase contains 4 iron sulfur clusters, and these clusters are important to catalyze the reaction. It does not participate. The iron clusters do not participate in the reduction as most of the iron sulfur clusters do, but it holds on to the serine group, the OH group of citrate to facilitate its elimination. So this step is not overly regulated as such. Step 3, we take isocitrate through the enzyme isocitrate dehydrogenase, and we end up at the end with alpha-ketoglutarate. There's an intermediate in this process, oxalosuccinate, that occurs. It's a decarboxylation of isocitrate to give alpha-ketoglutarate. Isocitrate dehydrogenase is definitely an allosteric enzyme. It's inhibited, this step is inhibited by ATP and NADH, and it's activated by ADP and NAD+. It's a very essential process to create alpha-ketoglutarate. Step 4. This is where we take alpha-ketoglutarate. We go to alpha-ketoglutarate dehydrogenase complex, and the result through that, and the result is succinyl-CoA plus carbon dioxide. It's a decarboxylation of alpha-ketoglutarate. The alpha-ketoglutarate dehydrogenase is also known as oxoglutarate dehydrogenase. It's a synonym. 2 carbons of the acetyl group, 2 carbons of the acetyl group of the acetyl-CoA are still present in the succinyl-CoA. So that's part of the process. This is a multi-enzyme complex. It is inhibited by ATP and NADH and succinyl-CoA, and it's activated by ADP and NAD+. This is a regular. So this is step 5. The 2 acetyl groups of succinate are now equivalent, and we are now going to take succinyl-CoA through succinyl-CoA synthetases the enzyme to make succinate and CoA, thiolated CoA. And we get a molecule of GTP. So often in this count as ATP, but regardless. All right, step 6. Succinate dehydrogenase converts succinate to fumarate. In this step, this is found in the cell membrane of bacteria and the inner mitochondrial membrane of mammalian cells. That's where the conversion of this occurs. There's 2 classes of enzymes because there's multiple active sites. The first class includes some succinate analogs, things like malleate and oxaloacetate, and the synthetic analog malonate, and they contain the strongest dehydrogenase inhibitors. The second class includes ubiquinone analogs, carboxin, thionyl trifluoroacetone, and that are other regulators of this molecule. Step 7, we have the hydration of fumarate to malate through fumarase. Malate is chiral, and it can exist as some enantiomers. It's produced in the cycle as a single stereoisomer. Fumarase is used in that cycle to conduct the transition step. It's a reversible reaction, and so it's very commonly unregulated. All right, step 8, oxidation of malate. Malate goes to oxaloacetate through malate dehydrogenase. Oxaloacetate can now react with acetyl-CoA to start another round, and that allows for the process to start all over again. Malate dehydrogenase, most known for its role in this cycle, and it's critical to the whole process as this makes it a cycle. So we're now back at where we started, and we can move forward. So here's the overall energetics of this profile, and you will note that we're producing quite a bit of intermediates for oxidative phosphorylation. I would know this overall reaction. Again, just to give you the idea, these things generally occur in the cytosol, but there are several parts that occur inside the mitochondrial space, and the little chart on the top shows that most of the cycle occurs inside the mitochondrion. This gives the control of cycle. There are 3 major feedback mechanisms. Citrate synthase, which is the first step, is inhibited by ATP, NADH, and succinyl-CoA, and it's inhibited by its product. Isocitrate dehydrogenase is activated by ATP and NAD+, inhibited by ATP and NADH. Alpha-ketoglutarate dehydrogenase is inhibited by ATP, NADH, and succinyl-CoA, and it's activated by ATP. I did mention several others, but these are the major ones, and so these are the ones that I would know for your exam. All right. Again, I just want to reiterate this chart right here is a great reference for you if you need to understand, and just as a reference point, and also for studying. Thanks for listening. Look forward to seeing you in class.